Hello, fellow foodies, and welcome back. This is Dr. Cassandra Quave, and you're listening to Foodie Pharmacology, the science podcast for the food curious. Today on the show, we're going to explore just how sweet nature can be. I have on the show a guest who is an organic chemist. His name is Dr. Grant DuBois, and he's a research scientist that focuses on non-caloric sweetening technologies. Grant began his professional career with Dynapol Company on sweetener research and then joined Cyva for immunoassay fluorescent probe work. He then joined Searle Pharmaceutical Company to look into sweetener research, which led to the development of Neotame. Then he joined the Coca-Cola Company, where he was named the company's first research fellow. This research led to the first zero-calorie frozen beverage, the first sweetener receptor positive allosteric modulators, and so were generally recognized as safe approval of the stevia sweetener riboside A. In 2012, Grant joined the stevia sweetener manufacturer Almedra as the CSO. Grant's Almedra research led to breakthrough in formulation technologies, markedly improving the taste of all sweeteners. Concurrently, he served as the VP of Research for Crave Crush LLC and developed geminic acid, a dietary supplement for the treatment of obesity and diabetes, which has been commercialized as Sweet Defeat. So as you can see, he's had a long and illustrious career with a lot of expertise in sweeteners. And so I'm really excited about this opportunity to ask him lots of detailed questions. Thanks so much for coming on the show today, Grant. It's great to see you. Great. So you're an organic chemist and your research is focused on sweeteners. I guess maybe we can start with a little bit of the basics about how sweeteners activate our taste bud receptors and how do they actually send these sweet impulses to our brains?
Sure. So actually understand very much about that until really the last 2 decades. And some phenomenal research done, enabled by the Human Genome Project, research that was done by a collaboration between Zucker at the U San Diego and at the NIH. And they really were the first to clone a sweetener receptor in rodents. And then Charles Zucker, with Lubert Stryer and Roger Chen, they founded a startup company called Six in the San Diego area to things having owned these receptors. They look to do high throughput screening and break new sweetener technologies. I was at Coca-Cola at the time, and we weren't interested in new non-caloric sweeteners. We had aware of thousands of non-caloric sweetener molecules that chemists like me had made, but none of them tasted like sugar. So the idea was, this activates the sweetener receptor, and that's what we really want, is the taste of sucrose. So how can we simulate the taste of sucrose with lower calories? And the idea came up, well, what if we find a fold or 20-fold enhancer of sucrose? Well, in medical chemistry jargon, enhancers are called positive allosteric modulators. And so we funded research at to find such enhancers. And we did. The problem was we could only find most efficacious enhancers we could find were twofold. So that would only enable us to reduce calories by 50%, which was not enough. I mean, for products like Coke was marketing, we needed to reduce calories by 10-fold, 20-fold. And being the case, we started to look at other technologies and continued to look for better enhancers, and they worked on it for about 20 years with some very capable people, but they never found enhancers that were more than about twofold. That got us into, at Coca-Cola, looking for non-caloric sweeteners that tasted more like sugar. And after getting all the possibilities of natural non-caloric sweeteners, I settled on a stevia sweetener called riboside A as the one that best met the metrics for commercial viability.
What can you tell us about the stevia plant? I have it growing in my garden. It's one of my it's one of my kind of just fun garden trick plants. I let people taste it when they come through my garden, and it's always surprising just how intensely sweet it is. So, how research really began on media? Was it always known as like a sweetener, other traditions, or how did we start working on this particular?
Yeah, well, it goes back quite a ways. The chemical structures of the sweet components in Stevia rhodiana first elucidated in the 1930s by many groups working on it. They worked out the chemical structures, but then um, people to really look at commercializing stevia sweeteners were Japanese groups, and it was very, very expensive in Japan, and they have always had an interest in natural products in Japan, and so they utilized stevia sweeteners early on, but they didn't taste very good. The early stevia sweeteners didn't taste very good at all, and so work was done to that problem. Innovation was at Almedra. Thought initially that if they could just get one of the stevia sweeteners called side A very pure, that would be the solution to the problem. We would have great taste, but not so. Plain taste, no real bitterness or anything like that, but this objectionable temporal characteristic with a little bit of a slow onset of sweetness and then a
Mm.
long lingering aftertaste that.
Yeah, it does have that aftertaste, doesn't it? Yeah.
And so joined, well, it's good before that, because when I was still at Coca-Cola, Coke marketing wanted to have a natural sweetener. And so, in all the possibilities, as I said already, stevia sweetener side A looked like the most attractive, considering all of the metrics for commercial viability, which include taste quality, safety, ability in water, stability, both hydrolytic stability and light stability, cost, and patentability.
It's
And
interesting because these are all qualities that we look for on the pharma side of research. Like in my own work that's translational, those are all the exact same factors we look for.
yeah,
So really, when you're looking for new food products, in many ways, it seems like it's similar to the drug discovery process.
yeah. So with the sweetener side A, when I was at Coca-Cola, we started looking for ways to try to formulate side A so that it would taste better.
Mm.
We didn't understand, no one understood in the early 2000s what caused this slow onset of sweetness and this long lingering aftertaste. And it was about that time that connected with a pharmacologist at Emory Medical School. His name's Kenneth Miniman, and you understand that this is probably due to nonspecific binding of these sweeteners to sites in the oral cavity. And with that hypothesis that that was cause, we started looking at ways to formulate it. But we really didn't make that much progress while I was at Coca-Cola. We did find that if you formulated it with erythritol, a natural sweetener you already mentioned, that we could make it much better. But erythritol is expensive, and if
Mhm.
you have enough erythritol in a beverage with side A to give good taste, it costs prohibitive. So
Yeah.
that's a practical solution.
Can
And
you back up one moment? Because I'm always thinking in my head, I have a lot of students that listen to the show, and they may not know what you mean by the term formulation. So, when you're talking about formulating a natural product into something that could be deployed as a beverage ingredient, what does that actually entail?
yeah, so you have to understand what your objective is.
Mhm.
And in our case, formulation of side A and other non-caloric sweeteners was directed toward trying to find a way to get rid of this accelerate sweetness onset and to this lingering sweet aftertaste. And so how do that? Well, we knew we'd do it with erythritol, and we also found that we could take basically any high osmolarity solution and it would taste much better. So it still didn't explain to us in the 2000s how to solve the problem. And it really was after I attended a seminar given by Professor Riddick of MIT. She was working on understanding how to formulate therapeutic drugs so that they would have more rapid bioavailability. And she explained that when therapeutic drugs reach the small intestine, what has to happen is they first have to diffuse through a layer of mucus, a mucus hydrogel that covers the entire alimentary canal from the mouth all the way down here. And so that was a big idea, because then I realized, oh, our whole tongue is covered with a layer of mucus. Humans secrete anywhere between 1 and 2 liters of saliva per day, and a major component of human saliva is a protein called mucin. The particular one in the mouth is mucin 5B, and it coats the tongue. So we've got about a 25 micrometer thick layer of mucus hydrogel that covers our tongue, and these sweetener molecules have to get through it before they get to the receptor. And sugars, being very hydrophilic molecules, go right through it like a shot, and they get away from the receptor quickly too. They diffuse away quickly.
Mm-hmm.
But these amphipathic molecules like riboside A and even aspartame and other non-caloric sweeteners, they get hung up in trying to diffuse through this hydrogel. They bind to every hydrophobic site as they go through, and eventually they get there, but they get there slower. And then when they
Okay.
get off the receptor, they have to diffuse away through this hydrogel again, and they get hung up, and they come back and activate the receptor over and over and over, and that's what creates this lingering aftertaste. So this is our new understanding based on Professor Ribbick's work, and the question was, well, how do you solve the problem? Well, it turns out that when you still have these still in your salivary gland cells, the glycoproteins are bound to calcium and maintained in a very compact form, very compact. But then once they leave the salivary gland cells and enter your mouth, they form this, they expand tremendously and form this thick hydrogel covering your tongue. And given that that's is happening, we understood that, oh, calcium ion binds to the mucus hydrogel and makes it smaller. So take a genius to think that, gee, wonder what would happen if we formulated any of these sweeteners with a calcium salt.
Wow. And
So that was
so
a eureka moment.
that's huge. That's huge. And so when you say that you're formulating, you're not actually changing the physical structure of the chemical riboside A. Instead, you're adding additional molecules to this mixture that's then being delivered. Is that right? Okay.
Exactly. And so I did the obvious experiment. I did riboside A in a system with chloride.
Mm-hmm.
Worked, but the problem was we had to use such high levels of calcium chloride that we also got salty, bitter, and astringent off taste.
I see. Okay.
And then, well, the periodic table, there's another mineral right next to calcium, that's magnesium, which is also physiologically important. We want
Mm-hmm.
to see what would happen if we used a mixture of magnesium salt and a calcium salt. And that was the big discovery. That a mixture of a magnesium chloride and calcium chloride, we're to use much lower concentrations, and now not get any off taste from the salts. And it would apparently create pores, or either it would cause the mucus hydrogel to shrink so that the sweet molecules could get to the receptor quickly and get away from the receptor quickly, or maybe it was creating pores. We don't really know. We haven't done the biochemical studies, although that's something that's underway now.
That's great. So for the average person, if they are, for example, opening up a packet of stevia to sweeten their beverage, the molecules or the molecule derived riboside derived from the stevia plant. So it's not like a plant extract, it's a single molecule. And then you have this calcium and magnesium mix. Are there any other kind of fillers or substrates in that mixture? How does that all come together? Because I know that riboside is a very potent, right, as a sweetener. So you have to really dilute the actual amount of the compound in a sugar packet. It's not quite the same as just putting a packet of white sugar. Maybe
Yeah.
you can explain that a little bit more.
Yeah, sure. If you go to the grocery store and go to the section of the grocery store where sugar is on the shelf and all sweeteners, you'll see all the choices. You'll see the pink packets, which are sweet and low saccharin type products. You'll see the blue packets, which are aspartame. You'll see the yellow packets, which are sucralose, and then you'll see the green packets. Most of them are green anyway, which are various stevia sweetener packets. And most of the stevia sweeteners that you have there, and you might have 5 or 6 choices of different stevia tabletop sweeteners. Most of them are formulated with maltodextrin or dextrose, and they really don't taste very good. It does taste very good. In fact, I think the only one that tastes very good is one that goes by the brand name of Truvia, is marketed by Cargill, and a blend of riboside and erythritol, as I said before, like many osmotically active materials, seems to have similar effects as the mineral salts in that I think it causes the mucus hydrogel to shrink, and the sweetener can get to the receptor and away from the receptor quickly, and it just works. So Truvia is the only one that I really recommend because it tastes pretty good.
Yeah. Well, and about these other sweeteners, we not just stop at aspartame? Like, why we keep looking for other sweeteners? Are there downsides to these other sweeteners in terms of flavor or health value?
Yeah, there are like 6 non-caloric sweeteners on the market. So-called artificial non-caloric sweeteners market in the world today, and they are saccharin, cyclamate. Saccharin was commercialized by Monsanto as its first product, actually, in 1905, and it was the only one for a long time. And then cyclamate was discovered at the University of Illinois in 1937, I believe, and was commercialized. And that enabled the first products, which were saccharin blend products, which were on the market in the 60s until after banned cyclamate for a decision that was not a good decision, but they did. Aspartame came along in 1981, and is, in my opinion, as somebody that's been in this field for 50 years as an expert, aspartame is the caloric sweetener, the synthetic non-caloric sweetener that I think is probably the most safe, in spite of everything one may read on the internet. And that's because it's just broken down in the gut to the natural amino acids, aspartic acid, phenylalanine, a trace of nothing. So that's aspartame. Then along came Sulfam K, developed by Searle Pharmaceutical in Germany, and then came sucralose, which was discovered at Tate & Lyle and developed by J&J. Since then, we've had neotame come along, which came out of the NutraSweet company, and Advantame, which came out of Ajinomoto in Japan. So all of these sweeteners are there. They're all looking for a piece of the action, but none of them replicate sugar taste very well because they all have this lingering sweet aftertaste.
Yeah. So do all of those share that kind of barrier of trying to get through that hydrogel on the tongue? Is that the main challenge across the board?
Absolutely.
Yeah.
And so our salt compositions that worded on in our June Journal of Agricultural and Food Chemistry paper works with all of them to eliminate the aftertaste. And the irony, or a serendipitous effect, which we weren't expecting at all, is when we first made these formulations of a mixture of magnesium salt and calcium salt, and later we added a potassium salt. When we first looked at these, all of a sudden there was this tremendous sugar-like mouthfeel.
Oh, mouthfeel is important. This is one of the challenges with the artificial sweeteners. Yeah.
Because even with sweeteners like aspartame, which have a very clean taste, you don't have any mouthfeel like sugar. And so there was some work done by Ajinomoto in the 10 years ago, and they discovered that the calcium sensing receptor, which is physiologically important in many parts of the body, also expressed by a subset of taste bud cells. And they reported that activation of the calcium sensing receptor on taste bud cells leads to a taste which they referred to as kokumi taste, a Japanese word which they translated as mouthfulness. And so we propose that our use of mineral salts, which are a mixture of a calcium salt and a magnesium salt, magnesium also being a known agonist at the calcium sensing receptor. We propose that these are activating a taste bud cell calcium sensing receptor, giving this pleasing mouthfeel. So,
Incredible.
yeah, it was
So
very fortunate.
what's next for, so tell us a little bit more about the paper. Maybe we can dive a little bit deeper into how you formulate this mixture, and then how do you test its efficacy? Because are these being tested, for example, in cell models or in animal models, or does it go straight to human testing? Like, how do we even detect sweetness in a laboratory setting?
Very good question. And this is predominantly done by human-based panel testing. And so what's done, and we have an expert panel, the technique is called descriptive analysis, and the panelists are trained to taste a solution and then report on the intensities they perceive of sweetness, bitterness, saltiness, slinger, and mouthfeel. And so they taste the sample and they rate all of these taste attributes on a 0 to 15 scale. Of course, since we're trying to eliminate sweetness linger, that's one of the most important attributes we look at, as well as the mouthfeel intensity. And involved in work while I was still at Coke, we were trying to develop a non-taste panel method for quantitating some of these things. And well, I'm a co-author on a paper that was done by some people, well, who's jointly at Georgia Tech and at using an fMRI technique where students are used to do an fMRI machine. They're doing functional magnetic resonance imaging to get excitation of neurons in the brain. And you're able to actually see that sweetness comes on quicker than aspartame sweetness. So,
Interesting.
so it can be done by fMRI, but that's a very sophisticated and expensive
Yeah.
equipment to be doing a lot of routine
See
work.
those things. Yeah, I mean, the assets that we have in my research group, we've done a little bit of collaboration with a company on looking for novel sweeteners, but the challenge is really having a high throughput assay. Saying it's kind of maybe one company has a kind of cell-based assay, but there are patents around that, and it's hard to, you can't really roll that out to other places for testing.
And the
Yeah.
first company to develop a cell-based assay with the sweetener receptor was Synomics in the Diego area. And
Mhm.
we funded, at Coke, we funded that work for about years. After that, Coke decided that this wasn't going anywhere. We weren't going to find positive allosteric modulators, or in other words, enhancers that were
Mhm.
twofold enhancers. And if we did, there were still going to be synthetic compounds. And we decided this was not going to advance what Coke was trying to do, which was more in the natural world, and we wanted tenfold enhancers.
Yeah.
Pepsi funded Synology for about 10 to 12 years after Coke exited, and they didn't find anything any better than we had, really.
Okay. Yeah, it's challenging, I can imagine, to find these. They have to fulfill so many unique criteria of cost, criteria at the level and like biochemical level. Well, a question about another natural sweetener. It's a bit unusual, but I'm guessing that you've probably heard of this. It's a molecule called miraculin, or it's a, from Syncytium, I think it's dulcificum. It's the miracle fruit, and people use this to flavor trip with, right? So you take this berry, you put it into your mouth, let it roll in the tongue, and then if you eat a very high acidity food right after, it makes that food taste intensely sweet. I do this in my class with, and then we have like lemon wedges, and they get to
Right,
taste that difference.
right.
Something like that, is that, has commercialization of that ever been explored, or is it kind of because a two-step process? There's just too many barriers because you have to pair it with a highly acidic ingredient.
Yeah, miraculin is a very interesting material, and there was a company in the Boston area in the late 60s, early 70s,
Mhm.
Maryland Corporation, that tried to commercialize it and put it out in the market, but the FDA shut it down because they wanted to see toxicology studies before they had to sell it, and so they went out of business. But miraculin's been around for a long time. You can buy it from, oh, there's a company, I can't remember the name right now, but there is a company that you can order it from over the internet. And it's a very interesting effect. You're right. It has no taste itself, really. But then if you taste anything sour after it, tastes remarkably sweet,
Hmm.
a very pleasant sweetness as well. I remember we had a party at our house, geez, 30, 40 years ago, and I had some Maryland, and people were drinking wine and drinking beer and things like that, which of course are somewhat low pH.
Hmm.
And so I invited them to a little bit of Milan and then taste a lemon wedge to perceive
Yeah.
remarkable effect. And everybody thought it was really cool.
Yeah.
And they went back to their bottle of beer or their glass of wine, and the wine and beer tasted sweet for about an hour. So it's a long-lasting effect. Yeah, maraschino is an interesting material, but no one's found a practical way to use
Yeah.
foods and beverages at this point.
That's great. Well, about, there's a lot of interest, scientific interest, a lot of commercial interest in these, non-caloric sweeteners now for, it sounds like, almost a century. If you're
Right.
going back to the early 1900s, what drives this interest in your experience? Is it really, to health value, or kind of what is it about these non-caloric sweeteners that is so heavily desired in the marketplace?
Yeah, I think it is a health issue. You may know, located right next to the CDC, and looking at the CDC website, the latest data I saw was that 41.9% of Americans are obese, at least
Yeah.
in 2020. And a very significant percentage of them are struggling with type 2 diabetes. So,
Mhm.
Americans are consuming, and not just Americans, but worldwide, we're consuming way too much sugar, high fructose corn syrup, and caloric sweeteners like that. But we have a insatiable looking for sweet tasting foods. And so, how to produce them without introducing all of these calories from or its equivalents? Well, non sweeteners have been the answer ever since Monsanto commercialized saccharin in 1905. And of course, they've been mainly found in caloric beverages. Uh, Increasingly, I think we're going to find that they'll be used in other foods, taking the sugar out and replacing the sugar with non-caloric sweeteners. And people are very interested in natural. And we've done some preliminary work. Well, our early work with Rubatioside A has been in beverages because that's
Mhm.
the big opportunity right now. But we've also done work in sugar-free baked goods, sugar-free frozen desserts, and so forth. And we found that preliminary experiments, we can make a cake or cookies with Rubatioside A and our mineral salt taste modulators. And instead of sugar as a bulking material, we'll use maltitol,
Mhm.
which doesn't provide much of a glycemic effect. And get cookies and baked goods and ice cream like products that actually preferred to the products made with sugar. They taste really good. And so I think this will be a big growth area in the future, enabled by these mineral salt taste modulators.
Yeah. Well, I mean, also, when you think about the American diet, most people understand that drinking a lot of sweetened beverages, that eating lots of ice cream and desserts is going to not be great for your health. But I
Right.
think one area where many people miss the kind of the ingredient list is when we go into other areas of the supermarket. For example, on our salad dressings, we have high fructose corn syrup. We have high fructose corn syrup in all of our sauces: barbecue sauce, ketchup, mustard, pervasive even in baby food. So we have these sweeteners that are just kind of into so many of our processed food products. You not even think of it at the moment that you're eating something that could be potentially unhealthy for you. I think it's easier to recognize, okay, yes, I should not have this extra slice of cake or this extra bowl of ice cream because maybe that'd be great for me. But people don't always think about that before they put their barbecue sauce on their chicken, or right,
Right.
or the canned tomato sauce, the pre-made tomato sauces for marinara sauce is also loaded with this.
Right.
So I guess my question is, do you think that there's a place for, this evolutionary drive for sweeteners? Do you think that there's a place for caloric sweeteners in other middle aisle products beyond desserts, beyond beverages? And Yes, is that going in terms of commercialization and research right now?
Yeah, I think there is a big opportunity, and I think these mineral salt taste modulators that we've been working with, they will make
Mm-hmm.
any of the non-caloric sweeteners, whether they be synthetic or natural, any of them taste better. If you tried to put Rebaudioside A just as it is into a ketchup or a barbecue sauce, it wouldn't taste very good. It would give you
Yeah.
this lingering aftertaste. And
Mm-hmm.
but in the presence of the mineral salts, I believe that have products that taste very similar to sugar and without the glycemic load induced by sugar or high fructose corn syrups.
That's great. So earlier you mentioned with challenges that folks face as they were trying to roll out products were toxicology tests. So for any new formulation, even for mineral salts, is there a barrier to commercialization that you have to go through? Basically, it sounds almost like what you would do for preclinical investigational new drug application if I were going to develop to be tested in the clinic. Is it similar to that? Like you need to have like animal toxicology results, or how get rolled out to the next stage of development?
Sure, good question. Well, come up with a new molecule like neotame was or advantame was. Those
Hmm.
molecules have to go through all of the toxicology studies that are laid out in a document published by the FDA called, generally referred to as the Red Book. And these
Hmm.
toxicology studies are very similar to what would be required for development of a new drug. The only difference, I think, is that clinical studies are not really required. It's basically all animal studies.
Okay.
Very expensive. It's very expensive. Not as expensive as a new drug, but work at the NutraSweet company, and product that came out of that was neotame. And
Hmm.
followed up with one of my toxicologist friends there, who said their best estimate as to the total cost of toxicology studies and related studies on neotame was about $83 million.
Wow.
So
Incredible.
significant amount of money, but not the billion-dollar type money that new
To get a
pharma.
new drug through. Yeah.
And there's another pathway to take a molecule to regulatory approval, and that's the GRAS process, generally regarded as safe process. And that's the process that we took to get Rebaudioside A for use in the United States by the FDA. And we still had to prove that Rebaudioside A, even though it's a natural product, and even though the indigenous people of Paraguay have been eating this material for thousands of years, probably, we had to do some toxicology studies. And
Hmm.
in this case, my best estimate was about $12 million because we didn't have to do we didn't have to do lifetime feeding studies because there was a Japanese company that had done a high quality feeding studies in rats. So we didn't have to do that, and it was less expensive. And now companies are using this GRAS process to commercialize regulatory approval for new sweeteners like monk fruit, and there are protein sweeteners like
Yeah.
brasine, and these are all being introduced to the market via the GRAS process.
Fascinating.
Yeah.
And so you've also had some experience with the dietary supplement approval pathways, or I don't know if there really is an approval pathway, but there is a dietary supplement market. Share with us about that work and how does that compare to your work in sweeteners?
Yeah, so the only experience I've had with that was with the company that I worked for a couple of years in the early 2013, 2014 timeframe. And they were interested in developing something that you could take a pill and it would block your sweet taste for an extended period of time. And it's long been known that acid, a material produced in this plant that's indigenous to India predominantly, would that. And we did develop material, a gymnemic acid, a purified gymnemic acid material as a dietary supplement. And it was introduced into the market, and it's being found useful for some people in losing weight because the idea is after a meal, if if you're fighting a temptation to choose a dessert, you could just take one of these tablets and it would make your ice cream taste like cold mush, or it would make it
So
taste like nothing
that's
interesting.
incredible. So it makes you basically helps to stop those cravings for sweet things because those sweet things now taste disgusting.
Exactly. Taste
Yeah,
good.
that's great. I like that. But you're right, it does come back to a traditional use of that medicinal plant that's known to have those effects on the palate. That's fascinating. I mean, there's a lot happening right now in the space for weight loss with, for example, with the semaglutide drugs and, gastric emptying as a strategy also for weight loss. I
Right.
think for many listeners, it's probably very confusing what all these different things, mean to our bodies because it's, pretty complex pharmacology. And even as scientists, we don't fully understand all the implications of semaglutide drugs.
Right.
Um, yeah. Um, but I think it's really interesting to see how organic chemistry can play such an important role in the development of new products and food products that can be tied to health and that helping reduce, of otherwise foods that could be harmful.
Yeah, I think absolutely right. Helps to have a chemistry point of view, but still it's critical to have a understanding of the biology of the system,
Mm-hmm.
at least as much as it's possible to understand it. As I mentioned earlier, in October of 1973, I started my first job with a company called Dynapol in Palo Alto, California. And we quickly realized that we have to solve this sweetness linker problem if we're going to have a non-caloric sweetener system that's going to taste good. And we tried all kinds of crazy things that were only enabled by the limited understanding that an organic chemist may have. And it
Yeah.
wasn't until the breakthrough in understanding the biological mechanism of sweet taste and the work on mucus by the Rivik laboratory that became possible to solve the problem. I have to tell you a funny story about work. I mean, DIPOL is an acronym for dynamic polymer. And the idea was to come up with food additives that would be safe because they would be non-absorbable through the GI tract.
Mm.
And so we were trying to make sweetener molecules that were high molecular weight so that they would be non-absorbable. And that we came up with a compound that's essentially non-absorbable, and it was sweet. The problem was when you tasted this material, there was zero sweetness for about 5 minutes. And then the sweetness started to build and build, and I couldn't rinse it out. You
Oh
couldn't.
no.
It built continuously for about 30 minutes or so. And then it took about 2 hours for the lingering sweetness to go away. And so realized that this was not a commercially acceptable strategy. We needed to find a way to come up with something that was safe, but also that tasted like sugar and not
Yeah.
have a tremendous sweet aftertaste. It
So
was
based on your years of experience, if you were to list the parameters of a dream molecule, a dream, caloric sweetener, what would those parameters be?
well, quality has got to be there. If you don't have quality, you're not going to sell it more than once. Got to have absolute safety. The regulatory agency will allow you to put it on the market. You've got to have solubility, good solubility in water so that it make added sweetened beverages with it. You've got to have stability so that it can stay on the shelf for a few months. You've got to have photochemical stability. Natural sweetener that did not is natural sweetener that comes from a plant that comes from South Africa. It's called monotin, and monotin is a tryptophan, substituted tryptophan type compound. And if you formulate it into a beverage and expose it to sunlight, it completely breaks down within
Oh,
a year or so. And the worst part is it produced a horrendous malodor. So
oh no.
you have to have light stability. You have
Yeah.
to have adequate cost also to address your market needs. And if you're going to spend all this money to commercially develop it, you have to have a way to get a patent on it so that you have some exclusivity for a few years.
Yeah, to recoup the
So that's
costs.
the common
Yeah.
method that we focused on.
Fabulous. Well, this has been fascinating, Grant. I think we're about out of time. For sharing your knowledge of this space. I learned a tremendous amount. I didn't realize, for example, how long research has been ongoing in this space on non-caloric sweets. I mean, century now. That's exciting.
Right.
And just seeing where things go in the future as we gain even deeper understandings of the taste receptors.
Very good. Thank you for inviting me.
Thank you very much.
Okay.
All right, foodies, you've been listening to Foodie Pharmacology, the science podcast for the food curious. I want to big shout out of thanks to our producers of the show, to Rob Cohen and Christine Roth. I also want to let you know you can find this and other notes on the show at our YouTube channel at Teach Ethnobotany. You can get a link to that and links to merch and all of our other episodes at the foodiepharmacology.com website. I'd love to hear from you. If you've got ideas on future shows or guests that you're dying for me to speak with, shoot me a message. You can reach me at social media sites. That's Foodie Pharma on Instagram or on Twitter or on Threads. We're everywhere now. So thanks so much for listening. Stay healthy out there, and I'll see you next time. Okay.